Welcome to Strand NGS webinar. We hope you're having a great day wherever you are in the world. We have a lot of registrants from everywhere. It's really exciting. So much. We are enthusiastic ourselves about Strand NGS. Today's webinar will be focused on fast and accurate variant calling in Strand NGS, but we also have a lot of new features. So I'm going to tell you a little bit about the new features, and then I'm going to jump right into the variant calling workflow. So what's new in 3.0? The very first thing you see when you boot up Strand NGS, and those of you who are using Strand NGS for the first time may not notice this, but people who've been using Strand NGS for a while will see that we've changed our welcome screen, right? So this is what you see when you start Strand NGS, both for the first time as well as every subsequent time. Whenever you close an experiment, you're going to see the screen. Whenever you start the tool, you're going to see the screen, right? And we're going to go over this welcome screen later on. For now, hopefully, it signifies the general changes in Strand NGS 3.0 as well. As far as the actual changes go, there's a bunch of them, right? So we start, and I don't know how many of you use snip effect analysis. I think it's a great tool. So in snip effect analysis, we've added HGVS notation. So I'll go over that really briefly. In ChIP-seq, we've added TSS plot. So this is the transcription start site plot. Wow, that's a tongue twister. But TSS plots are sort of, as the distance from the TSS increases, what's the coverage like? So that's a TSS plot. I'll go over that real brief. And we also have faster RNA alignment as well as support for circular genomes. But the major changes in Strand NGS all have to do with DNA-Seq, and so I'll be spending the bulk of my time in this presentation on DNA-Seq. Just to sort of recap what happens in DNA-Seq, you start with an input pass, which could be a paired end or a single end sample, right? And you go through alignment, you go through sort, and you remove duplicates, and you do some kind of local realignment or local reassembly, and then you do variant calling. And this generates your variant call set. Typically, we use the VCFs nowadays, so we just going to call our variant call set of VCF. And so when you go over all these steps, there's likely to be, issues and artifacts introduced by each stage. And so you have to benchmark the accuracy. And that's what we went ahead and did in 3.0. And we found that, our accuracy numbers are fairly good and sometimes very, very good. And I'll go over this in detail later on. We also doing really good as far as time goes. You can see here that this compares Strand NGS to the GATK best practices workflow that includes BWA as well as Picard tools and GATK for haplotype caller. And you see that we are about twice as fast as the GATK best practices workflow. And finally, our storage and visualization is also pretty top notch. So what we have here is on the left side, we have a graph that shows you how much storage you actually use up after the sort stage, right? And you can see in red, Strand NGS is pretty much a constant storage type phenomenon, whereas GATK is more, it's more linear as the number of stages increases. So I'll go over that in some detail. And finally, we have some excellent visualizations, and a lot of these visualizations have been around for a long time. And so essentially, DNA-Seq now in Strand NGS 3.0 is fast, it's accurate, it's storage frugal, and it's visual. So I believe that these are the sort of 4 selling points of Strand NGS 3.0 DNA-Seq, and I'll talk about that in a lot of detail. And we also put all of this into one best practices workflow. You should think of this as similar to or analogous to the GATK best practices workflow. So this is essentially, you could see that this is every stage in the workflow, right from creating an experiment from FASTQ files all the way to VCF. And this takes care of all the parameters and all the intermediate stages for you, so you don't have to actually worry about any of that. And this gives you highly accurate variant call sets from paired end DNA samples. So I'll go over the new features first. So these are the sort of this is a broad overview of the new features. There are a lot of other new features. I recommend that you look at our reference manual, you look at our release notes if you want to know more. But first of all, let's just look at TSS plots in ChIP-seq. Now, TSS is just a transcription start site, so where the transcription starts, and then the TSS plot just tells you what the coverage is like a certain distance away from a TSS, right? So this is in base pairs on the x-axis, and then you have coverage on the y-axis, and this tells you just how much coverage you have in reads per million, so many bases downstream or upstream. The important thing we want to emphasize about this is that this actually supports multiple samples and entities. It works exactly the way you expect it to work, and it works right out of the box. So hopefully our ChIP-seq audience will be very interested in TSS plots. RNA alignment is just faster. We went ahead and improved both transcriptome and transcriptome plus genome alignment. You should see these types of speedups. If you were using 2.9, if you were using RNA 2.9, you should see speed up factors of anywhere between 50% and 150%. And one thing that I'm sort of excited about is how we've added HGVS notation. HGVS has been around for a really long time. And we've just gone ahead and incorporated HGVS and SNP effect analysis. So we have 3 types of HGVS, of course, and you can see the protein, cDNA, and genomic HGVS. So for instance, over here, you have a transcript, right? And you want to know how a particular SNP, in this case, it's a CT substitution, and how it actually changes, the resultant amino acid. You look at the protein HGVS, it tells you, oh, this proline goes to serine over here. You can probably see that. I'm going to zoom in real quick. You can see that over here, P.Pro 243 serine. So that tells you exactly all you need to know about that particular SNP. So this is in SNP effect analysis. And of course, there's 3 different types of HGVS. Again, we support HGVS right out of the box. You don't have to worry about it. It works exactly the way you think it will. And just another thing for some people who might be interested in bacterial genomes, some of which are circular. We're looking at E. coli here, right? And there's a few things that are interesting about this. First of all, we've added circular genome support. It's been around for a while. It's not new to 3.0, but I thought I'll just briefly look at it because it's interesting for reasons that you may not really be aware of. So one thing is that you note that we have a circular genome here, right? So the final coordinate in the circular genome is about 4,641,000. You see that, right? And then we go right back to the start of the genome, right? And so this is called an elastic genome browser bookmark. So it actually spans the end of a genome to the start of you can actually combine the two. You could combine the end with the start. And in the circular case, the end is the start, right? And so what this also shows you, when you actually go ahead and browse, you can actually type in this as an EGB, as a command, and open up the EGB, which is the Elastic Genome Browser. And you can actually see that the coverage pretty much stays the same, which is a good sanity check for if you're actually aligning reads correctly to circular genomes. If you weren't aligning reads correctly, then you'd have all kinds of strange coverage artifacts right here at the boundary. So we've been supporting circular genomes for a while. We thought people who work with bacterial genomes might be interested in this feature. So that's as far as new features go. I mean, I recommend that if you're interested in our new features, you should definitely read the release notes. You should definitely read, the relevant parts of the reference manual if you're interested, and obviously get back to us for questions if you have any. I'm going to devote the rest of this webinar to how we actually achieved fast and accurate variant calling in Strand NGS. I'm going to give you a bird's eye view of DNA-Seq in Strand NGS. I'm also going to give you some sense of how we actually benchmarked Strand NGS, DNA-Seq in Strand NGS 3.0. So first, you saw these graphs before, right? We have accuracy, we have speed, we have storage, we have visualization, all of this stuff in Strand NGS 3.0 DNA-Seq. So how did we get here, right? What are the steps we took? And I'm not going to tell you everything because that's too long and there's just a lot of stuff in there, but I am going to give you a brief summary. I'm going to give you a few glimpses into how we actually, achieved all these things, all these great benchmarks of performance. So I'm going to start with something really simple, mapping quality. Some of you may be aware that when you create an alignment experiment in Strand NGS, you actually have something called alignment QC. QC stands for quality control, right? So you can actually click on alignment QC. You'll go to a list of tabs that are displayed over here, and you can click on each tab, and it will tell you, give you a histogram and so on. We're looking at the mapping quality tab right now, and those of you who've been using Strand NGS before have been used to our earlier mapping quality, which ranged from 0 to 254, and this one ranges from 0 to 70 now to sort of maintain better compatibility with other aligners like BWA and Novoline. But the more important thing is that mapping quality has been rigorously scoped out right now. It's a function of suboptimal matches, so it's both the number of suboptimal matches as well as the scores of each suboptimal match, the alignment scores. So this formula is actually in the reference manual if you're interested. So that's one big thing about mapping quality. The other big thing is that when you have a mapping quality of 0 now in 3.0, it just means that read with a 0 mapping quality has more than one optimal match. And we figure that you see that here, for instance, we have 2 million reads, almost exactly 2 million reads. This is a whole exome sample. So we have 2 million reads with more than one optimal match, and these have been assigned a mapping quality of 0. We believe that these are substandard reads that should not be included for variant calling, which is why we assign a mapping quality of 0 to them. And accompanying this change in mapping quality definition is a cutoff in SNP detection, which you see on the right, which actually tells you at what threshold to ignore a read with mapping quality less than, right? So right now it's set by default to 20, which not coincidentally is actually the same as the GATK cutoff. So we find that this is sort of an optimal sort of trade-off between false positives and true positives. So I'll come to that. But the idea is that the changes in mapping quality are also accompanied by corresponding change in variant calling as far as cutoffs go. The next interesting change from earlier versions of Strand NGS is the gap percentage. Now, if you wanted to detect long gaps in earlier versions, you actually have to, so here on the left side of the screen is the alignment parameters wizard. And if you wanted to detect large gaps, you would actually have to set the percentage gap to something like 40 or 50, right, depending on the length of the indel you were interested in. We've gone ahead and removed that completely. So now it doesn't really matter what you specify here in the gap percentage, because we've added a secret sauce to split alignment. So split alignment not only now detects the split reads, it also detects reads with really long gaps. These are both insertions as well as deletions. So you can probably run it with 2% gap or 3% gap, and it'll still probably give you pretty much similar results with all the long indels in there. So basically, percentage gaps don't matter as much in 3.0 as they did in earlier versions. In fact, in most cases, they don't matter at all. And you can imagine that this actually improves the performance quite significantly, because we don't have to do large gap percentage type things for too many reads. So for those of you who are interested in zygosity, I have good news. Realignment is great. So I'm just going to go over. I'm just going to go over a quick example of an insertion and how we are actually improving the zygosity of the insertion. So first of all, what is this insertion? It's a 4-base insertion. The insert sequence is CCCT. You can see the qualities here as well. This, by the way, is the genome browser. A lot of you who've been using Strand NGS for a while now are probably familiar with the genome browser, right? So this is a CCCT insertion with these qualities. The qualities are relatively good, right? And this is the location, right? So there's 2 read lists over here. The top read list is the read list before realignment in the top rectangle. And the bottom read list, you see it says here local realigned reads. These are read lists. This is the read list after realignment. Now, if you looked at only the top read list, you would probably and go, well, this is homozygous insertion to me, right? Because there's lots of reads here that support the reference. But after realignment, you actually look at it and say, well, wow, look at that. All the insertions line up really neat, and it looks like a homozygous one now, right? So if you never did realignment, you'll probably think, yeah, this is probably het. And if you did realignment, you'd be certain that it's a hom. So another way of looking at this is to use the variant support view. The variant support view, by the way, we talked about it. We showed it to you briefly in an earlier slide. I'm going to show it to you again. What is the variant support view? First of all, here's the reference on the top. You have CG, CC, etc., right? And on the bottom, we have 2 clusters of reads. The first cluster reflects all minuses at these variant locations, so that that means it just supports the reference, and it's of size 17. So that means there are 17 reads that are supporting it. And the second cluster has 15 reads supporting it, and is of course the CCCT insertion that we saw earlier. So the variant support view before realignment. And if you just look at this once again, you look at it and go, it's a het, because it's nearly more than half the reads support the reference. Now, after realignment, you see that you only have one cluster of any node, and that supports the insertion, right? And this is clearly now a hom, right? So 31 out of 32 reads support the insertion. So this is just another way of visualizing what we saw earlier using the variant support view. Now, how did this happen? This isn't magic. So what the technology that enables the correct zygosity here is placing the reads approximately, right? So you see that we have some of these reads here that are sort of clipped. So this part is clipped. You see that there's a cross hatching over there that may or may not be visible. I'm going to zoom in. Hopefully that's visible to you. So you see this sequence here is cross hatched, right? That means that it doesn't really align over there. We don't really know where that part of the read aligns, but we're pretty sure that this read belongs over here, right? That's the approximate read placement before realignment. Now, realignment takes this approximate read placement and actually says, well, I have 15 reads supporting this insertion. Can this also support that insertion? And the answer in this case is yes. And there's a whole bunch of these kinds of soft script reads, these approximately placed reads that go ahead and support the insertion. So the answer here is, why do we change the zygosity, and how did that happen? Well, we did have more evidence over there, and we now know how to incorporate the evidence correctly with realignment. So we achieve better zygosity through better approximate read mapping. That's the takeaway, really. And we believe that this is one of the many reasons that you should definitely do local realignment, especially if you're interested in indels, and especially if you're interested in calling the right zygosity. Finally, we have a few new cutoffs in variant calling. You already saw one of them, the mapping quality cutoff. I'm just going to go over them in some detail now. This is in the SNP detection tool. Just as when you click on SNP detection in the tool, you'll see this window here open up. And then you click on the advanced tab, and you'll see all these options. And in there are three options. The first of which is the mapping quality cutoff. We already discussed that. What is the rationale of the mapping quality cutoff? What is the rationale of any cutoff? The rationale is that if a variant is strong, then it has strong support from normal reads. Right? So what that's saying is that, if we have to call a variant, then it had better have support from good reads, i.e. normal reads. And in this case, normal reads have high mapping quality. That's one way of looking at it. There are many other ways to look at normal reads as well. The next way is the tail distance. Right? So set up this tail distance cutoff, which is also new in 3.0, is saying is that, well, let's ignore supported towards reads. So here's an example of that. So this is a TA homozygous substitution, right? The 7 supporting reads over here, right? And if you go ahead and actually look at the tail distances of the supporting reads, you have sort of falls in. This is a very classical picture of read support, right? The tail distance kind of falls, and then eventually you have no read supporting that variant. So 5, 4, 3, 2, and 1. It's just the number of base pairs from the mismatch to the end of the read. Now, what I haven't shown you is that there's more of the read that goes to the right side for each of these reads, and I've truncated those. So the tail distance is really measured from the left over here, right? So let's say the tail distance cutoff is 2, then these 2 reads go away. You ignore them as support, and you only include these reads when you're trying to call the TA substitution. So here the rationale is, once again, I'm going to repeat the same sentence. If a variant is strong, it should have strong support from normal reads. Now, in the tail distance cutoff, the definition of normal is a normal read specifies variance at least n bases away from either of its edges, where n is the tail distance cutoff. Finally, we have a split read filter. Again, in this advanced tab in variant calling. The rationale for this is actually very simple. Here's an example of a split read in the elastic genome browser. This spans 300 base pair deletion, right? So it's not a very long deletion, but it's not a very short one either. So what do I mean by not very short? It's not contained within the read. It's not very long in that it's not sort of hundreds of millions of base pairs away or in another chromosome. Right? So it's a moderately long deletion. And there's some support for it, right? But what do we also see over here on the left? We also see some stray mismatches on these split reads. Now we want to ignore these mismatches. Some of those mismatches may actually line up and cause some spurious SNPs to appear, right? And we want to ignore those because, again, the rationale, if a variant is strong, it should have strong support from normal reads. In this case, we are defining a normal read as not being split. Another way to think about it is that split reads already specify the split haplotype. In this case, the split haplotype is a 300 base pair deletion. And it's highly unlikely that they will also specify some sort of local haplotype. In this case, the local haplotype would just be a single base substitution. So together we have 3 cutoffs. Together, these cutoffs decrease the false positive rate, and you can feel free to actually change these cutoffs. We recommend the default values, which is why they're default. We believe that they work well for whole exomes and whole genomes. We benchmarked them on those. I'll come to that in a bit. But of course, you can set them to 0 if you don't like them. Set them to any value. These are our recommended values. So that was a bird's eye view of the kinds of changes that have gone into 3.0, the kind of changes in DNA-Seq that have gone into 3.0. And I'm going to talk a little bit now about how we're benchmarking this entire workflow. But we need to really understand what this workflow is before we try to benchmark it, right? So I'm just going to recap the workflow once again for those of you, who may not be familiar with it. So we start with the FASTQ, the DNA sample of any kind. It could be a single end, it could be paired end. And you do alignment first of all, right? You map it to the genome. Right? That's the biggest and most important, most time-consuming, most computationally intensive step, right? And then after that, you have to sort it. You have to remove duplicates. As you saw, we have to do local realignment. And then finally, you do variant calling. You generate a VCF. So how do we benchmark this incredibly complicated workflow? Well, there are ways. A lot of you may be familiar with this, but the idea is to start with some kind of baseline. And the whole exome baseline that we used was the GATK best practices workflow. We just took the input FASTQ, we subjected it to the BWA plus GATK workflow that a lot of you may be familiar with. I'm not going to go into that now. Right? For the whole genome, we actually have an even better baseline. We have a genome in a bottle baseline, right? And the genome in a bottle, I should recap for those of you who may not be familiar with it, is, it's a call set by NIST, right? The National Institute of Standards and Technology by Justin Zook. It has gained a lot of traction in the past few years as being a truth set for the 12878, who's a Central European female, right? And it's been published and available for a long time, and it goes through iterations like 3 or 4 times every year. So, and it's an intersection of 5 to 7 different types of workflows, right? So it's likely to be a lot more reliable than if we just did GATK best practices. Of course, we don't have the same kind of truth sets for other individuals. So our baseline for other individuals is just the GATK best practices workflow. So what do you do with this baseline? The assumption with the baseline is that everything inside the baseline is a true positive. Everything outside the baseline that's not in the baseline is a false positive, right? That's a very strong assumption, but that's the kind of assumption you have to make if you want to benchmark DNA-Seq workflow. Right, so and then we have the Strand NGS call set, which is VS. And then we intersect the baseline call set with the Strand NGS call set. And we can talk about how we did this if you're interested. But the intersection is not straightforward either. And then we generate false negatives, true positives, and false positives. What are false negatives? What are false negatives? They're ones that were captured by the baseline but were not captured by the Strand NGS call set. False positives are the opposite, those that were in the Strand NGS call set not captured by the baseline. The true positives are the intersection. Right? And again, I have to emphasize here that this is a fairly strict definition of accuracy. And there are many provisos that go into this. We can talk about that. So once you have the true positives, the false positives, and the false negatives, you can actually measure the so-called precision and recall. Precision is the measure of how many false positives you have. The recall is a measure of how many false negatives, so ones you didn't call, right? So, by the way, this is a great way to start thinking about accuracy, simply because if you call, hypothetically, if you had a SNP caller that called every location on the genome, then the false positive rate would be immense. You'd have a precision of 0%, but you'd have 100% recall because you would be guaranteed to call every SNP that exists, and vice versa. The challenge with any DNA-Seq benchmarking is to get both these numbers up to very high levels. And that's exactly what we've done. And we have a white paper out on it. And if you're interested, you can definitely read it, right? Essentially, we have now accuracy as far as precision and recall goes for both whole exome and whole genome samples for both the entire call set as well as the subset of indels that are very high, right? And that really encourages us, right? And I have to emphasize here, you just want to go into this in a little bit more detail, to emphasize here that every step here these figures were obtained after every step was native to Strand NGS, right? We never used a third-party tool at any point in time, right? So all these steps, so the cumulative inaccuracy of all these steps is at worst measured by these figures over here. So hopefully this gives you a lot of confidence in just DNA-Seq within Strand NGS. Hopefully this tells you that you can use the entire workflow and that it's reliable. But if you're still not convinced, we have some other figures to convince you. First of all, you might think, who's using Strand NGS? We are actually the biggest users of Strand NGS in-house, right? Apart from the customers we have all over the world. And this exact same workflow has been accredited by the College of American Pathologists as well as the NABL. And our clinical team uses DNA-Seq in Strand NGS. We have 3,000 samples that we process every year, right? So we eat our own dog food, is what I'm saying. So that's the first reason to be confident. The first other reason to be confident. But I'm also going to tell you what I already told you, which is that we are twice as GTK best practices. So once you have accuracy figures, go and actually measure the time, right? You do alignment, you do everything, right? And so we are twice as fast as the GTK best practices workflow, right? And really, the other big thing is that we have really small storage footprints starting from the sorted BAM, right? Up until the sorted BAM, you have to generate some sort of read list. But after the sorted BAM, right, we use very little storage. So imagine you have 20 whole exome samples, right? There are stages over here, or 5, however many stages there are after alignment. If you use something like a GTK practices, you're using 20 times 5 storage, whereas in Strand NGS, you're going to be closer to 20 times 1. There's some variations and the graph reflects that. It's the broad picture. And the final case, I think, for doing everything within Strand NGS is that the results here are incredibly structured. There's a huge hierarchy here. It tells you exactly what you did, how you arrived at a particular BAM, right? And often when you're looking at just a file on the hard drive, you don't really know how you arrived at it. But if you're within Strand NGS, you don't have to worry about that. So that's the structure. That's what the structure goes. And we have really good visualizations, and a lot of you using Strand NGS probably attest to this. But I just want to show you the variant support view really quickly, right? So here's a SNP location, right? And it has 1,000 reads that cover it, right? And then if we actually click, and you already saw this earlier, the variant support view, but I didn't show you the other stuff that's inherent to it. You can actually split up the clusters by strand. So that's an instant picture right there of strand bias, right? And you can also, this entire grid can be colored by base quality. The variant bases are obviously marked out, right? And you also have the size of each cluster over here on the right side. So these are things that are incredibly useful to the biologist, the bioinformatician, right? And we believe that it'll be useful to you as well. So I want to just, and that concludes our DNA-Seq benchmarking effort, presenting that. And I just wanted to really quickly go over how we've actually packaged this in Strand NGS. We have now something called a one-shot pipeline. This is in the welcome screen. This is the first thing you see in Strand NGS. It's also the first, it's also the screen that you are presented when you close an experiment. So this is sort of what you return to. And right there on the very top left corner is the one-shot pipeline. So when you click on it, right, there's a couple of intermediate stages, but you'll actually be led to Strand NGS best practices. And you probably saw this earlier in the talk, but this actually goes over every single stage in the best practice workflow. All the parameters inherent to each workflow. I think there's probably something like 40 parameters here, and each parameter, there may be some categorical ones, but a lot of them are numerical. So you don't have to actually worry about figuring out what each parameter is. The best practices actually does that for you. So if you're getting started with DNA-Seq for the first time, we highly recommend that you just use the best practices and see what kind of VCF you get, what kind of calls you get, what kind of indels you get, right? This works right now only for parent samples. So yeah, I mean, if you have parent samples, we recommend that you go ahead and try DNA-Seq best practices. That's most of our talk. This is Strand NGS. If you want more details, a lot of you know about all this already, but we have a great sales team and a great support team. We had a lot of great reviews about our support as well, so please contact them. Please contact sales, right? And I'll be happy to field your questions. Thank you so much.